Good morning, everybody. My name is Travis Van Leeuwen. I'm a research scientist with Fisheries and Oceans in Newfoundland and Labrador. I'm in the Salmon section. We also have Mark Young on the call. He is from the Department of Fisheries, Forestry, and Agriculture. And I just want to emphasize that this is a project that was really run by the province. It was funded by the province, and it was really in-kind support from DFO. Obviously, we don't have a lot of time here today to go through the whole study, but if you're interested, this study, the results of this study has been published in the primary scientific literature. That's this long-term survival Atlantic salmon following catch and release considerations for anglers, scientists, and resource managers. And that's in fisheries management and ecology. And then for those of you interested in a longer version, 50,000 plus word version with lots of details, there was a release of a report about this study as well. So you can check that out on the province website. And also there's some videos there and things like that that just sort of summarize the results of the study. So again, in the interest of time, just a quick overview of the presentation today. Going to go through a little bit of background, methods, going to do those things very quickly. Spend a little bit more time on the results. Obviously, this is kind of the bread and butter of the study. And then we'll get into some discussion. And if we got time, we'll talk a little bit about future directions. Obviously, a lot of data generated throughout this study. And what you're going to see here is really just the survival evaluation of that. And so for decades, For decades, catch and release fishing has been used by regulators as a management tool for the recreational Atlantic salmon fishery. Obviously, as was the there's a lot of debate among anglers regarding the effectiveness of catch and release. And, there's nothing new here. This debate's been around for a long time. And a lot of this debate really comes down to demographics of anglers. So anglers have, different, values in terms of the fishery. And so depending on those values, some anglers like, retention fisheries. They like to retain their fish. That's the sole reason they go out and fish. Some people like to catch lots of fish. Also, some anglers just like to be out in nature and just spend time. And so there's that side of the debate and the ethical animal welfare side of the debate. But then there's also some debate in and amongst the scientific literature. And a lot of that debate within the scientific literature is driven really because of different techniques. So to date, there's upwards of 20 scientific papers focused strictly on anadromous Atlantic salmon and catch and release. And the techniques used to evaluate, catch and release has differed. Some of them are laboratory-based studies where they use simulated catch and release angling. Some of them are field studies. Some of them are done in Norway, Scotland, Ireland. And of course, they have different regulations associated with their fishery. And so, those differences, whether they're allowed to use barbed or barbless hooks or, bait, lures, those kind of things, they trickle into, our understanding of catch and release and really the applicability of these results from other studies in terms of what it means for our fisheries here. We have very restricted, strong regulations. So single barbless hooks, artificial flies only. And so, really some of that debate comes from, the applicability of these results. And so if you're interested, there's a couple of other papers from our group over the last kind of couple of years. One kind of synthesizes all that literature into one manuscript, and that's the mortality of Atlantic salmon after catch and release angling, assessment of recreational Atlantic salmon fishery in a changing climate. And there's also another one related to catchability and water temperatures. So feel free to look those up. Hold on here. Now, the purpose of this study is really in two themes. So the first part, and it's really broken down into 4 parts, but actually 2 themes. And so the first part is to compare long-term survival between Atlantic salmon that were caught and released to a control group. And we're able to follow fish from 1 to 300 days. The second part of that was to determine if mortalities associated with catch and release occur before or after spawning. And then another part of that is compare the number of fish that overwintered and migrated downstream to sea as kelp. And then really the other theme of that is to really, try to understand the mortality that occurs and what's driving that mortality. And so really taking it a step further and trying to understand provide fishery-specific survival rates for cotton-released Atlantic salmon across water temperatures. And as we all know, there's lots of debate around environmental protocols. And so, we wanted to spend a portion of this time actually trying to do some applied science. So the study was taking place at Western Arm Brook. That's on the island of Newfoundland. It's up on the northern peninsula here. And there's a number of reasons why this site was chosen. There's data collection here since the early 1960s. There's a counting fence in operation since 1971. So we got lots of really good biological data on this. The fish tend to be one sea winter maiden spawners. They average anywhere from about 45 to 74 centimeters. And the sort of population on average is about, almost about 1,000 fish return to Western Arm Brook every year. And really a kicker to this is that it's actually closed to angling. And so it provides a really unique opportunity to have, somewhat of our own unique experimental stream to do this work. So obviously, when we're assessing survival, we needed some way of following fish or, tagging fish and following them to assess survival following, the control groups and the experimental groups. And so we used, again, two groups. So one was a control. These are fish that were collected at the counting fence. So they were dip netted out of the counting fence, tagged, and then released. And then there's, of course, our experimental group. And so all fish were essentially handled in some capacity. And then, the experimental group had the added stressor of the angling event that occurred on them. We used low-tech radio telemetry tags, and we used a combination of external and internal radio tags for this study. And there's a couple of reasons for that. External tags, this is a picture of them here. The lifespan is about 5 months, so it's a relatively small tag. And then internal tags have a lifespan of approximately 2 years. And so, there's a lot of discussion around, the possible side effects of doing surgery, anesthetic, and things like that. So we wanted to really bring in two aspects of this study. One, we wanted to have a look at a tagging procedure was very quick. And so that's the external tagging procedure, didn't use anesthetic. And then, but also we wanted to incorporate some of the internal tags just so we can get some longer term data on these fish as we go forward. So here's just a few quick slides of the tagging procedures. So again, for the control fish, we had 164 control fish, and for the angled fish, it's 119 fish, and that was over the course of 3 years. So for the external tags, fish were dipped, they were measured, they were tagged, and so that radio tag was actually attached to a flow tag that people are probably fairly aware of, and then those fish were released. So really a quick procedure. No anesthetic, sometimes less than a minute for that procedure to take place. So this is just a picture of what those external tags look like. So here's a flow tag, just in the dorsal musculature of the fish. And then you have the streaming antenna of that radio tag off that fish. Just to give you an idea, the difference between an external tag and an internal tag and the sort of complexities that come with internal tags. So fish were dipped. So whether that was at the counting fence trapped or whether the fish, the angled fish were brought to shore, they were all dipped with a knotless rubber net. They're placed in a cooler with clove oil. That's the anesthetic that we use. And then the tags were implanted via abdominal surgery. Most people are probably aware of this type of procedure. It's done a lot with Atlantic salmon tracking studies. They were held until they're recovered and then they were released. So here's just a few pictures of that, internal tags being inserted into the fish. And again, here's the procedure taking place. And here's the difference between the internal and external fish. So you can see the tag's been placed internally, and then you have this external antenna that comes out of the fish. So to monitor those fish after they were released, there were 7 solar powered telemetry stations. There's also manual tracking that occurred, and then also 49 telemetry monitoring flights occurred in helicopters. So really extensive monitoring of these fish throughout the study. And so here's just a quick schematic here. I don't want to spend too much time on it, but here's the control fish and the angled fish. Here's the layout of Western Arm Brook where the study took place. The red triangles just show the extent of the radio receivers. So these are those fixed stations. So you can see they're pretty spread out through the watershed. On the experimental figure here, angling locations were essentially fixed. So they occurred, over a very small stretch of river. And then in both, you can see the counting fence down here. So all fish had to pass the counting fence, except the angled fish had the extra stressor of the angling event. So we're actually controlling for handling effects. Besides the kind of standard air temperature, cloud precipitation, those kind of, bits of data, there's a number of other various aspects of data collected during this study. So things like, weather location of the fish, hook location, hook time, land time, handling condition, and then everything associated with the different surgeries and procedures with the tagging. And I'll just talk about those real quick here. So this is not really anything that, this is just showing some of the various bits of data that people might be interested in on this call. In no way is it controlled for effort, but it's something to consider in terms of the study. So here in panel A, number of salmon and water temperature. So the average temperature for this study was about 15.6 Celsius. Most anglers were experienced anglers. However, we did have a range from, say, less than 5 years to greater than 20 years of experience angling. Again, this is not controlled for effort, but most fish were caught between 8 and 10 a.m. And then you have some angling that occurred 4 to 6 and 8 to 10. But, angling did occur and fish were caught throughout the whole day. In terms of where fish were hooked, so after those fish were netted, most of the hooks actually fell out. So that's something to keep in mind. There's a whole slew of different places within the fish where those hooks ended up. Everywhere from the upper jaw, right jaw, those kind of things. Some a small fraction were actually foul hooked or hooked in the tongue. Now, before you go and throw away all your flies, one of the common responses from anglers when we asked them, what fly were those fish caught on? A common response was none of your business, and then followed by a blue charm is sort of second into that. So. Don't go throwing away all your bombers and things like that. It doesn't necessarily mean that blue charm caught most of the fish because this isn't controlled for effort. So, it's just a common fly people are using. So it's just to give you an idea of the data. And then in terms of different data associated with the different procedures, most fish on average took 4 minutes to land. Again, we have quite a bit smaller fish here in Newfoundland. And, different times for the anesthetic to knock the fish out, average about 5 minutes. Surgery recuperation, about 12 minutes. And so for those external tags, like I said, it's a very quick process. So from the time that fish is dipped to release is about 4 minutes. And then with those internal tags, because of the anesthetic process and the complications that come with that procedure, the average time is about 23 minutes before that fish is released. Now let's get into some of the data here. So, this can be a little bit tricky sometimes to interpret, but I also want to just acknowledge a couple of things. So obviously the angled fish is a group here, and that's, that group stayed throughout the whole study. So it's basically an average of the 3 years. And we ended up generating 2 control groups because what we learned very early on in the year 1 external controls is that some of these individuals that were handled with cotton gloves, we started noticing some marks and sores on those fish as we tracked them through the system. Now, it's unclear if those marks and sores were, they're obviously associated with the cotton glove in some aspects. So whether it's the cotton glove rubbing off the slime of the fish and making them vulnerable to infection, or whether it's a combination of the cotton glove and the handler holding the caudal peduncle too hard. Now, what's important here is that this is year one of the external controls. Again, it was a 3-year study. And again, not all fish, it was, we were unable to, visually inspect all fish after release. So not, it's unclear whether all fish handled in this group developed the marks and sores. But what we do know for sure is that not all fish died. And so what we wanted to do, instead of, manipulating the data in any way, we wanted to just generate, 2 sets of controls. One included those external cotton glove handled fish, and one excluded them. Now, it's not as simple as that, because when you exclude those cotton glove handled fish, obviously that's one treatment group from 1 year. And so you start bringing in other effects that could affect the interpretation of the study. So when I go through the study, there's, the data is presented a couple of different ways. So let's get into the data real quick. So again, this is just a summary of the data over the 3 years. We have survival probability on the y-axis, days of the experiment basically on the x-axis. The control fish, so panel A again includes the cotton glove handled fish, whereas panel B excludes the cotton glove handled fish. And so what you see here is the control fish is that pink line or that red line there, and the angled fish is the blue line there. And again, we were able to track fish from 0 to 300 days. And you can see that the confidence intervals overlap between the control and angled fish. And that occurs in both, whether you use the differing controls. And so the overall conclusion is that there's no significant difference in long-term survival between Atlantic salmon that were angled, radio tagged, and released and the control group. And again, from those figures, we can actually generate some survival probabilities for individual fish. And so we arbitrarily took, set days to do this. So 1, 10, 30, 50, 150 days, and 300 days. And you can see if we just take the simple average of those time durations, if cotton glove handled fish are included, we get a 98% survival relative to the control group. So that's suggesting that the added stressor, the catch and release event, accounted for 2% of that mortality. And then if the cotton glove handled fish were included, you can see that the survival relative to the control group is 94%, so 6% mortality. And you can see that, consistently with both groups, survival is very high throughout the study in both groups. But it's important to emphasize that, the average temperature here was 15.8 Celsius. Now, that's great, 1 to 300 days, but we wanted to put this in some sort of biological context. Obviously, the fish that we're dealing here, we get a lot of the 1C maiden spawners. Anywhere from 10 to 20% of these fish will survive to actually head back out to sea. So they'll overwinter and head back out to sea. So we wanted to put this in some sort of biological context because a lot of these fish die naturally in these rivers. And so I don't want to get too hung up on the various figures. Again, panel A, cotton glove handled fish included, panel B, cotton glove handled fish excluded. And so what we see, if we look at the number of fish that survived up until November 30th, this is generally the time when spawning has taken place. We can see that 70% of the angled fish survived beyond November 30th, so that's the spawning time, whereas 59% of the control group of known fate survived the spawning time. So basically we conclude that there's no difference in the amount of fish that likely survived to spawning time. And the same with cotton glove handled fish excluded. So we get 70% of the angled fish and 67%. So no difference in the amount of fish that survived up until spawning time. And then we were also able to evaluate kelp survival. So, was there any difference in the amount of fish that overwintered and came back down the river past the counting fence? And so for the angled fish, that ranged from 9 to 10%. And for the control fish, again, whether cotton glove handled fish were included or excluded, that was in the range of 13 to 19%. Now, we have to remember that sample sizes, at this time, were relatively low. So that difference of say 9% equates to maybe 2 or 3 or 4 fish. So really, again, no real difference in overwintering survival between the two groups. So next, we sort of evaluated survival. And so we wanted to really get into the nitty gritty of, we know that some mortality occurs. So what's really driving this mortality? And so obviously, where a lot of the other research is focused has been around things like hook type, bait type, air exposure, those kind of things and they look at, the effects of that. Now, given the regulations here in Newfoundland and Labrador, single barbless hooks, artificial flies, and also assuming that anglers are using best practice, really what stands out in our study is the effect of water temperature on survival of fish. Now, this is an important consideration. Obviously, it leads into things like environmental protocols and things like that. So I'll just walk you through this study, this analysis here. So on the y-axis, we have survival probability. On the x-axis, we have water temperature. And again, we pulled 1 day, 10 days, and 30 days post-release. Black line indicates the angled fish. And then the red dotted lines indicates the 2 control groups. And so you can see that with the angled fish, whether it's 1 day, 10 days, 30 days, we see a very strong negative relationship between survival probability and water temperature. So as water temperature goes up, the survival probability of fish goes down. Now, what's also interesting here is that after 10 days and 30 days, we can see that the control group somewhat converges with the angled group. So what it means here is that the effect of water temperature on survival probability is relatively short-lived. So either that fish survives or dies. And that sort of binary response happens within 1 to 10 days. And so really, when we're talking about environmental protocols and survival of fish relative to the control, it's really, 1 day post-release that we're looking at, because you get a significant difference between the angled fish and the control fish. And so let's push that a little bit further and just look at some of the numbers. Again, there's 2 ways of looking at this data. And so there's been some confusion over the last few months here related to this data. And so one is looking at just strictly survival probability. So, we take an angled fish, we look at the survival probability, and we say, what's the survival probability of a fish post-release, 30 days post-release, and we can give you that number, right? But really what we're talking about in terms of the added stressor of catch and release is what is the relative effect associated with the added stressor of catch and release. And so where we see that difference is 1 to 10 days post-release. And so these are the numbers that we generate here. And so you can see, again, this is where a lot of the discussion happens with environmental protocols, anywhere from 18 to 20 degrees. And so you can see that the added mortality, depending on which control you use, cotton level handled fish included or excluded, at 18 degrees, anywhere from 7 to 8%, from 19 degrees, anywhere from 9 to 11%. And then when you start getting into 20 degrees, it's anywhere from about 13 to 15%. Now we have to keep in mind all these fish are handled in some way, but at least this starts putting numbers around individual survival probabilities of fish. Now I know this is kind of where the literature is kind of focused on in terms of explaining environmental protocols, but what we need to remember here is we need to be moving beyond individual survival probabilities and start looking at, what does this mean in terms of population numbers? What does it mean as a whole in terms of catch and release fisheries? And so in our previous paper, we touched on this. And what happens in Newfoundland and Labrador is that most of the fish enter our rivers kind of mid-June to sort of early July. So we get 80% of the run moving through during those periods when water temperatures are cool. Now, when environmental protocols start kicking in and water temperatures start increasing, we actually start seeing a very strong, there's not a lot of fish that are actually being caught at those temperatures. And so whether that's a combination of environmental protocols taking place, whether it's anglers not going out fishing during that time or switching activities. And so when we start putting these numbers to population type analyses in our rivers, we start noticing this equates to maybe 1 to 3% of the population that are actually dying as a result of catch and release. So really low numbers. So more of that kind of discussion is in any one of those papers I mentioned earlier. And so just to sort of recap, results consistent with previous studies that suggest high survival Atlantic salmon following catch and release. Results reiterate that with strict management restrictions in place and those currently used in Newfoundland and Labrador, in addition to salmon handled properly, that survival following catch and release is high. And the importance of water temperature, in addition to salmon handled properly, in ensuring the survival probability of Atlantic salmon should not be underestimated. And so just some future directions that are coming up from this work. We kind of finished with the survival part of it. And we're going to be looking into things like movement and behavior analyses, pre and post or indirect effects of catch and release. So we have some work we have some additional sampling in the works right now where we're hoping to reassign parentage to smolts that should be heading out of the river in the next, this year and for the next 2 years. So we can identify some possible fitness related differences, or at least look at that. We're going to incorporate some field surveys, GIS photo interpretation and techniques to characterize differences in habitat use post-release, and then overlay of salmon locational data to determine key migration and holding zones in the river. And so with that, a number of acknowledgements. This was a huge effort. So again, in-kind support from DFO, the Western Arm Brook County Fence staff, the province fish forestry and agricultural staff, and again, Canadian helicopters.